So let's at first talk about the protein pathway synthesis. The formation of saliva is a protein synthesis, storage, and then stimulated exocytosis. At first, the start of saliva formation in the endoplasmic reticulum of these cells, the control of the production within the endoplasmic reticulum comes from the DNA in the nucleus. Okay, so the pathway at first, these will be transported from the endoplasmic reticulum via the Golgi apparatus to the transphase of the individual cell, and then moves out of the cytoplasm of the mucus or the serous acini. The apical end of the cell will accumulate granules of future saliva as an initial storage, and within these immature granules, a concentration of secretory proteins in later on mature secretory granules. Exocytosis will be then of the stored proteins into the lumen of the acini, and from out there these secretions will move to the duct system, the intercalated, the striated and intralobular duct cells. Here is the luminal surface and here is the apical surface of the protein secretory serous cells. These are the intercalated intracellular canaliculi in between, and we will talk about the vesicular protein pathways in the parotid acinar cells. So number one will be storage and storage granular pathway. Number 2 and 3 and 4 are constitutive pathways. So the saliva is constitutively moved to the apical membrane, or number 3, or to the basolateral membrane in number 4. And number 5, we have transcytosis from basolateral towards the apical and membrane of the cell. We have also noted that the serous cells, following the synthesis of protein secretory products, they will be either resting unstimulated serous cells containing large number of secretory granules in the luminal parts, or they will be the stimulated part, with the stimulation upon mealtime, upon mastication, upon also will be severely depleted during night, the number of granules. Here we go for parasympathetic and sympathetic neuro innervation, fiber innervation of the acini that helps in saliva production. We have embryologically transmitters that cooperatively influence the neuro peptides and neuroeffector sites. We have either number 1 epilemmal. A is epilemmal, remaining outside of the basal lamina, these neurotransmitters, or they can be these sites beneath the basal lamina, in direct contact with the plasma membrane, and these are called hypolemmal. So as a conclusion, the parasympathetic affection efferent innervation will form the cause formation and secretion of the granules and the fluid by the secretory acini, while the sympathetic drive will usually increase the output of the secretion. Both of these pathways will cause the contraction of myoepithelial cells that help in these myoepithelial cells help in the direct fluid to move from the acinar lumen along the duct system. This slide shows an electron micrograph of the myoepithelial cell. See, with multiple processes. This surrounds the secretory acinar cells and the intercalated duct system, the smallest duct system. You can see there's a flat nuclei. In the dendritic processes, we may have cytokeratins and actin filaments. Hence the prefix myo, it means a muscle. It has the ability to contract like a muscle. These projections will surround the acinus, and when this myoepithelial cell will contract, it squeezes the acinus to secrete the saliva and helps it move out of the duct system. Note, these are the secretory granules in the serous cells. Returning back, this is the parotid gland showing secretory parenchyma in these parts, and which is divided by lobules, divided into lobules by this septa of stromal connective tissue that you see up here. So you can see also interlobular collecting ducts. You can see sometimes arteries, lymph nodes, and plasma cells, and fat cells, of course. Once again, this is the intralobular part from the parotid gland, where you see the serous cells, only one type, and these are the small intercalated, but mostly between the intercalated and striated ducts. The striated ducts have the more columnar cells, and they surround a bigger lumina. Here are the darkly stained serous acini, the parotid gland, and you see the smallly squeezed intercalated ducts which open into a bigger striated ducts. If we see a magnified look of this intercalated opening into a striated duct, we can see some change of the shape of the cells. Also, you can note the myoepithelial cells around the acini and around the intercalated ducts, which squeeze the fluids out. You can spot some here plasma cells, or fatty cells that are not obvious in between the acini. Also, we see the serous acini of the parotid, the intercalated ducts, which open into larger lumen striated ducts. Also, you can see the parotid gland showing intercalated ducts leading from serous acini into larger striated ducts. Note that the cuboidal epithelium cells lining the intercalated duct have prominent nuclei, and the nuclei in the striated ducts are more centrally positioned. So the saliva is firstly secreted from the acinar cells, and then resorption of NaCl will occur in the striated duct cells to transfer the saliva from isotonic to hypotonic saliva. This is a higher magnification of the intercalated to striated ducts. In the striated ducts, you will have active transport of the ions, Na, potassium, chloride, and carbonate. So if you look at the intercalated duct, we have cuboidal lining cells and we have small amounts of intracellular organelles which are associated with protein synthesis, while in the striated duct we have the abluminal, both the laminal and the abluminal surfaces are brush surfaces with mitochondrial enfoldings that involve the active transport of ions. Also here you see intercalated ducts leading from serous acini to striated bigger ducts. The white spaces are fat cells. Remember, we have high number of intercalated ducts in the parotid system, and the striated ducts also are longer and more branching. These are the striated ducts, and of course more active in protein synthesis. Here we see a view of the striated duct. Small intercalated ducts are not so obvious, but you can spot the presence of plasma cells. Darkly stained serous acini parotid gland, and you see bigger and more obvious columnar organization of the cells around the striated ducts. Multiple fat cells can also be seen. See the brush border of the striated ducts. Also note the large cytoplasm and the large central located nuclei. Microvilli are all on the surface, the luminal and the basal surface. That's why it's called a striated duct. Moving to a very lower magnification, we can spot the collecting or interlobular ducts. They have more columnar pseudostratified appearance. These ducts have unstriated luminal and basal surfaces. We have a layer of basal cells, and we have the main mucosa and surrounding connective tissue adventitia. Again, a more closer look for the pseudostratified appearance, columnar cells. Near the surface, these cells will become stratified to mingle with the outside orifice, opening into what's called the Stensen's duct outside the oral cavity.